Here's a cell. This cell is diploid. That's because it has 2 copies of each chromosome. Here are chromosome number 1, there's 2 of them, and here are 2 chromosome number 2s. Therefore, we say that this cell has a chromosome number of 2n. Now, this cell can go through 2 different types of cell division. It can go through mitosis or it can go through meiosis, depending on which type of cells and which type of reproductive style the cell needs to use. So we're going to compare those 2 processes right now. So let's take a look at our original cell. Each of our original cells, whether they're in mitosis or meiosis, are going to begin in interphase and the S phase. If we look at the cells in interphase, they're each going to have 2 copies of each chromosome. These are called homologous chromosomes. That's because the chromosomes, this chromosome here and this chromosome here, carries the same kinds of genes. Same thing here, this chromosome and this chromosome carry the same kinds of genes with the same information. Now, during the S phase, these chromosomes get duplicated, and the chromosomes are held together by the centromere. We call these sister chromatids. They're twins. They're exact copies of each other. So now, once the interphase and S phase is completed, we move on to prophase. Now, prophase and mitosis and meiosis are pretty much the same, except that in meiosis, our chromosomes form a tetrad. Our homologous chromosomes form a tetrad with all 4 homologous chromosomes. Also, we have crossing over in meiosis, where a piece of DNA from one chromosome swaps with a piece of DNA from another chromosome. This introduces additional genetic variation. We do not see crossing over in prophase in mitosis. Next is metaphase, where the chromosomes are going to line up across the metaphase plate, the middle of the cell. In mitosis, each individual sister chromatid lines up across the middle, whereas in meiosis, the tetrads line up across the middle of the cell. During anaphase, the chromosomes are pulled apart. In mitosis, this process is going to be used to separate the sister chromatids, so we get 1 copy of each chromosome on either side of the cell. In meiosis, the goal here is to separate the homologous chromosomes. So here's one, here's the other, moving towards opposite ends of the cell. Then during telophase and cytokinesis, the cells split apart. So after this step, we have 2 cells in both mitosis and meiosis. Now, if we look at mitosis, this process has been completed. There is 2 copies of each chromosome in the cell, and that process is finished. But if we look at meiosis, we still have our double-stranded chromosomes, which we do not want. So meiosis is going to go through all 4 stages again. So here's prophase, okay, where the chromosomes are getting organized. During metaphase 2, the chromosomes line up along that metaphase plate in the middle of the cell. During anaphase, here is where we actually separate those sister chromatids, so they get split apart, broken in half, and move towards opposite ends of the cell. In telophase and cytokinesis, our cells are going to split and form 4 cells, each with one set of each chromosome. So now this process is complete. Let's compare the 2 processes. Here's our original cell. Remember, our original cell started out as diploid. It had 2 copies of each chromosome, 2 short chromosomes, 2 long chromosomes. If we look at the products of mitosis, those are still diploid. They have 2 short chromosomes, 2 long chromosomes in each cell. And if we look back to our original cell, we can see that these cells are identical. They have the same amount and kind of chromosomes. If we look at the products of meiosis, these are haploid. Their chromosome number is 1n because they have 1 copy of each chromosome in them. We can see they have 1 short and 1 long. 1 short, 1 long, 1 short, 1 long, 1 short, 1 long. And we can see that each of the cells that we created are very different from our original cell. There's genetic variation that has been introduced with meiosis. Hope you enjoyed the meiosis-mitosis comparison.